This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. So here in the 5th chapter, Principles of Inheritance and Variation, see how characters are getting inherited or how it is passing from one generation to another generation. That's what we are learning in this chapter. The main concept or the content of this chapter is that. So in this chapter, mainly we are learning um, like what is this inheritance or the concept of that inheritance. Then Mendel's law. Mendel's laws of inheritance we are learning, and inheritance of one gene, how that single gene is getting inherited. In that, under that single gene inheritance, we are learning about law of dominance, law of segregation, then exceptions of that. Everywhere, see, if you are learning about one concept like Mendelian principle or laws of inheritance by Mendel, there also some exceptional cases will be there. So that's what we are learning, exception to Mendelian principle. Then inheritance of two genes, how two genes are getting inherited. Like when from single gene, we are moving to the concept of two genes, how that inheritance is taking place. In that law of independent assortment, we are learning. Then two genes, how they are interacting with respect to that post-Mendelism. Okay, then how sex determination is happening, then what is mutation, cytoplasmic inheritance. um, so all these concepts, these are the main contents of this chapter, Principles of Inheritance and Variation. Okay, let's move to this part. So in this part, like we have already discussed about the reproduction of all organisms, right? So while reproducing, a progeny is receiving the characters from parents, right? A progeny, see, this is the parental plants. From there, the progeny or in the next generation, plants are getting, or the offsprings are getting, or that progeny is getting the characters from the parents, right? So how that characters are getting passed in the form of from male parent through sperm it is coming, and through in female parent through egg it is coming, right? And both are joining together, that is called as fertilization, and after fertilization zygote is forming. So there will be a blend or mix of all these characters, parental and maternal and paternal characters, right? So for example, if you are planting a mango seed, that is giving only a mango plant. The baby plant will be a mango plant, right? It won't become a jackfruit or something else, right? So exactly the parental characters it is representing. And in the case of animals, an elephant will give birth only to the baby elephant, right? It won't be a dog or cow or something else. So this is called as inheritance. So the progeny which is produced, that resembles the parents, right? Resembles, there is a resemblance. Okay, we can't say exactly it is like the parents, all the characters are not like the parents, but it is, it's not identical in all the way, it is having resemblance, right? And the resemblance we said, there we need to mention the variations also. What do you mean by that variation? See, here both these characters are joining together, and here the gametes are formed and plants are there. Here four plants you can see, right? Each plant will be different. See, if your parents, they have three children, think about them, each and every one will be different, right? There won't be, like, even in the case of twins also there will be similarities, but along with the similarities, differences also will be there. Or those differences are called as variations. So this variation, degree of variation in each progeny or in each one, here 4 plants you can see. In each plant, the degree of variation differs. It won't be identical. That degree, here 1% difference is there, means in another plant it may be 4%, or here it may be 5%. See, in the first plant, more paternal characters are there. It is showing more. See, in our home, just consider in our home, 3 children are there. Among them, first one will be like they'll be having dad's features. Like if they have dad's features more, like the skin color or hair, nature of hair, then height, everything matching to father's character. In the second, it may be that second may be more similar to mother. So that degree of variation, that is also different in each one. Okay, so here, so in the previous parts we have learned about Mendel's law of inheritance. So like these characters are getting inherited, right? So John Gregor Mendel, he was the first person who demonstrated the scientific basis of this inheritance and variation. How he proved? From parental, this is called as hybridization from parental plant. Here you can see this is a parental generation, and from that he made F1 generation. So in the F1 generation, he said that all the plants in the hybridization experiment on P, which he conducted, in that he mentioned that he selected some characters, and based on that he made his study. So in that he saw in the F1 generation, generation genotype all were tall, right? All were tall only. When they came to the second generation, here in F2 generation, they can see tall and dwarf plants, right? So here monohybrid cross. Here from this experiment, single pair of contrasting character. That's what he considered as a trait. So that is called as one gene inheritance. Okay, one gene inheritance. So what Mendel did, he crossed true breeding tall variety. True breeding in the sense both, like both the alleles, the concept of alleles, and we have already discussed that alleles, both the alleles are similar. Okay, it's like similar alleles are present. That's why we are calling as true breeding tall variety. He crossed with true breeding dwarf one. Okay, so the true breeding dwarf variety pea plants, he crossed with true breeding tall variety pea plant, right? So the plants which is used in the initial cross, that is called as P1 and P2, parent 1 and parent 2, P1 and P2. Okay, so this pea plant, you know, that is self-fertilizing. Okay, so before conducting the experiment, the anthers should be removed from the female parent. Why? Before maturity, we need to do this in order to do the cross fertilization. Pea plant is self-fertilizing, right? So we need to remove the pollen to conduct the cross fertilization or cross pollination. So the method of removal of anthers from bisexual flower of female, that is called as removal of pollen. So we are removing anthers from the bisexual flower. Bisexual flower means both pollen and anther both are there in that flower from the female parent. From female parent, right? So from the female parent, we are doing that is called as emasculation. So that is the emasculation. So the pollens, when these pollens are at the stage of dehiscence, that from the male parent that will get collected and again that will get dusted on the stigma of that emasculated flower. So in that emasculated flower or in that female flower, the pollens are removed. So for fertilization, they need the pollen which we need. The character we are going to give another parent's character. So we need those pollens and those pollens are get collected and dusted in the stigma of emasculated flower. Then Mendel collected the seeds which are produced as a result of this crossing. Cross pollination, then collection of seeds. Okay. So after collecting the seeds, this is the procedure. So first these P1 and P2. Then from that, which type of gametes you will get? Same. This is tall one, this is dwarf one. And those, if you are dusting the pollens from the male flower, which you have with the desired character in the emasculated flower, female flower, then this is the condition, tall one and dwarf one. Both will join together here, mating will happen and that will produce, see, now we are in this stage. Cross pollination over, seed formation also over, and that seeds Mendel collected. Then that seeds, next what he did, he collected the seeds and they produced next generation, okay, which are produced through the cross pollination. Those seeds he grew and he produced the first hybrid generation. This is F1 generation, first filial generation. That's what he produced. This is the seeds collected. In those seeds, this will be the condition. Okay, capital T and small t. And he grew those seeds and produced next generation. That is called as F1. Okay. And in that generation, in F1, see after collection of seeds, what he did? F1 generation. So in the F1 generation, Mendel found that in F1 he saw, see how that F1 was. This is tall. Here one dominant allele and one recessive allele. Here you can see, right? So this is tall. Here also the same, one dominant and one recessive allele. So here also the dominant allele character will get expressed. So that also shows tall. So in the F1 progeny, all plants were like one of its parents, right? Which parent? It is like the tall parent. It is resembling the tall parent, right? So none were dwarf. All were showing tall character. So he made with other characters also, he conducted the same experiment in the same way, right? Like stem height he considered like tall and dwarf, then flower color. So he considered different characters, several characters he considered and repeated the experiment. So in that also, he got the same result like flower position, pod shape, pod color, seed color, all these characters he considered and repeated the experiment. And in everything, he got the same result. Like only in the F1 generation, he got the same result like the parent or the F1 progenies which he produced that were showing or that were showing similarities to one of the parent. Then after that, what Mendel did, this F1 he allowed for F1 he allowed for self fertilization. Okay, then he conducted self fertilization, let them cross themselves, let them get pollinated themselves, like that he left. So the same, in the same flower, both male and female parts are there. So from that, pollen grains will fall on the stigma of the same flower. So that is called a self fertilization, right? So after that, that seeds also he collected and made them to grow. Okay, this is self fertilization here. And he raised F2 generation. So in the F2 generation also, what result he got? In the F2 generation, offsprings were derived from selfing of the F1, right? Here self1 plants he made to F1 plants he made for self fertilization, and that is called as F2 generation, which is the second filial generation or F2 generation. Okay, so here the plant proportion was like 3/4. Here you can see among these four plants, 3/4 of plants. 3/4 means one, two, total four plants. Among that, three plants. Out of four, three plants were showing tall character, right? And out of four, one plant was showing dwarf character, right? This is only one is dwarf, remaining three were tall. Okay, here what we need to notice, here the character which is not present in the F1 generation, which was that character, the dwarfness. Here only in F1 you got only tall plants. Mendel got only tall plants. But in the F2 generation, that character came. The disappeared character, the character which did not express in the F1 generation, that got expressed in the F2 generation. So here, dwarfness which disappeared in F1 generation, that reappeared in F2 generation. So the tall and dwarf traits in F2 generation were identical to their parental type, and they did not show any kind of blending. That means all the offsprings were either tall, here you can see either it may be tall or dwarf, right? None of the offsprings, here you can see either it may be similar to the tall parent or it may be like the dwarf plant. So none of the plants, none of the offspring showed a blending of character. None were in between height. Okay. So here with the example, we try to prove that involving a single pair of contrasting trait or contrasting character, they conducted a cross, and this is called as monohybrid cross. A single mono, here you can see single character they considered and they did hybridization. So there is no blending of character, either it was similar to tall or dwarf. Okay, so tall and dwarf parents both got crossed and generated F1. Then F1 allowed for selfing, then produced F2. In that, 3/4 were tall and 1/4 dwarf. Okay, so with the same method, he made similar crosses with other pairs of contrasting traits also, and Mendel observed that F2 generation in which both the traits were expressed in the proportion of 3:1. So the phenotypic ratio is 3:1, right? 3:1. Like three were tall, one was dwarf, right? That is a phenotypic, how it is getting expressed, the expression of that character. Out of four plants, three were tall, one was dwarf, right? So that's how he proved monohybrid cross. This is how Diagrammatically, we will represent the monohybrid cross. Okay. Then here, before going to the next topic, just to make everything clear, we need to explain some more points like concept of factors, right? So Mendel did these experiments. So based on these Mendel's observations, okay, so Mendel proposed something is passing down without any change from parents to offsprings through gametes, right? Pass something is passing from where to where? From parents to offsprings, right? So through which one? Through gametes. It is passing. So this is happening over successive generations. He called these things which are passing from parents to offsprings, he called these as factors. Okay, so that is called as factors. So now we are calling these factors are called as genes. Now it is called as genes. So therefore, a gene, what is gene? Gene is defined as the functional unit of inheritance. This is the functional unit of inheritance. Okay, so a gene is called as the functional unit of inheritance. And these genes, they are having the information which is required to express the trait. If you're considering a particular feature like flower color, those genes which are carrying that particular character, they'll be having the complete information to express that particular trait in an organism. Okay, so chemically, these gene, that is the part of DNA or segment of DNA, and that will be doing a particular function. Okay, they'll be synthesizing the polypeptides for that particular character. Okay, and genes which are coding for a pair of contrasting character, contrasting trait, that is called as alleles, right? Actually, these alleles are slightly different forms of same gene. Alleles, we are saying, here you can see two alleles of the same gene. Actually, this allele is representing the character tall, or the tall feature that is represented by these two alleles, right? Capital T, capital T. Here both alleles are similar, but this is representing the feature tall, right? But these alleles, whatever we are calling, that is actually not similar, slightly different forms of same gene. Alleles are slightly different forms of the same gene. Actually, the gene, how we are representing this? We are using alphabetic symbols for each gene. So capital letter, this capital letter we are using for the trait which is expressed in the F1 stage, and small alphabet for the other trait. For example, here we are considering the capital T that is used for tall trait, and small t, small t is used for dwarf, right? Then here capital T and small t, capital T and small t. These are the alleles of each other. So in plants, a pair of alleles for height, how we can tell that? It may be capital T or capital T or small t, then small t. All these are representing the height of plant, right? But we are not using capital T for tall character and D for dwarf character. Why? Because these are the different forms of same gene, and one is representing the dominant character, another one is representing the recessive character, right? Then homozygous and heterozygous condition. Homozygous and heterozygous condition. Actually, in a true breeding plant, tall and dwarf P varieties. Here you can see the tall and dwarf P varieties are there. So there is allelic pair of genes for the height. In tall one, that allelic pair is capital T, and in dwarf one, small t. This is the allelic pair, right? This is tall and this is dwarf, right? So this condition that is termed as homozygous. Okay, who coined this? Like the scientist named Bateson and Saunders. Bateson and Saunders, they termed as homozygous. Homozygous. Zygous condition. Here, both are similar. Capital T, capital T. This will produce gametes, capital T and capital T. Right. This will produce gametes, small t and small t. See, from this, it will never, okay, it will never give one capital T and one small t. Right. Because there is no capital T allele. So different alleles, they are called, here same alleles are there. That is called as homozygous. But if, see, think about the F1 which they produced, Mendel produced, that is in the condition of capital T, small t, one parent, and another parent is also in the condition of capital T and small t. Right. If it is giving gametes, how it will be? One is capital T and another one is small t. Okay. So that condition which are able to give two different alleles, that is called as hybrid. This is the hybrid condition. Both characters are present, right? So that is a hybrid condition. So like this, if they are able to give different types of allele, that is called as heterozygous condition. Heterozygous condition, and Saunders termed this condition as heterozygous. Saunders, he termed as heterozygous. Clear? Next, what we are learning is genotype and phenotype. Okay, genotype and phenotype. See, genotype means the genetic representation. Genetic representation. Genetic representation of an individual uh, with respect to one character or more than one character. That is called as genotype. See, here we are representing capital T, capital T. This is the genetic representation of tall plants, or otherwise capital T, small t. This is also representation of a feature. See, this is also tall, but here capital T, small t, small t. This is dwarf. So all these representation that is showing the representation of a character which is called as height. Clear? So genotype means the representation of genetic condition of an individual. Okay. And phenotype means the observable condition. Observable condition or the morphological appearance, that is called as phenotype. So this phenotype of an individual, that is determined by the combinations, right? From this feature or from this character, different combinations are coming, means here capital T capital, both dominant alleles are coming, means it will show the tall character, and one capital T and one small t, that also shows tall. But if another allele of the same character, tall, if it is coming in recessive condition, that gives a dwarf plant. Correct? So that is the observable condition of an expressed allele. Okay. So the tallness and dwarfness, that is the observable condition. Then the dominant and recessive, we know that based on the result of this F1 generation, Mendel, he was able to propose that two dissimilar factors are present in a single individual, right? Only one is able to express at a time and the other won't be able to express. So the one which is expressing, one which is getting expressed one is called as dominant, right? Right? The dominant factor, which is failing to express, that is called as, which is failed to express, that is called as recessive factor. Clear? So in like we can say that dominant allele that will influence the appearance of the recessive one, influence the appearance of the phenotype. The phenotypical expression is getting influenced by the dominant allele. Even an alternative allele is present, like that may be the recessive allele, even that allele is present, then also the dominant allele can influence the phenotypic expression, and that's why only the dominant character is getting expressed. See, here you can see the tall and dwarf, both alleles are present, tall allele and dwarf allele both are present, right? Here you can see that tall allele and dwarf allele both are present, even though the dominant character, which is that tall, that is getting expressed. The phenotypic expression, that plant is in tall character, or it is showing tall character. That means it is having, the dominant allele is having the capacity to express in the generation, or that can influence. Okay. So next we are moving to Punnett square. Actually, see these many things which I have explained, like dominance, then the uh, characters, all these which I have expressed, or just now which I have explained, that is for the detailed study of this Punnett square. Otherwise, you won't get the concept of the Punnett square. And if you are learning with this Punnett square about these concepts, it will be easy for you. Okay. So Punnett square, actually the production of gametes by the parents. The gametes are getting produced by the parents, right? So what will be the gametic condition? Half the number of chromosomes. In gametes, chromosome number will be half. But in parents, how it will be? Double, 2n, right? So the formation of the zygote from the F1 and F2 plants, that can be understood from the diagrammatic representation, which is called as Punnett square. So Punnett square is used to understand a typical monohybrid cross, like how Mendel conducted the true breeding tall plants and true breeding dwarf plants. To understand such a type of cross or such a typical monohybrid cross, Punnett square is very important. So geneticist Reginald C. Punnett, who developed Reginald C. Punnett. He developed this Punnett square and named after that. So from this, he calculated the probability of all the genotypes or all the offsprings in a genetic cross. Okay. So the possible gametes that are here, we are considering the tall plant, homozygous tall and homozygous dwarf. Okay, so which are the possible gametes? From the tall plant, homozygous tall, you will get two dominant gametes, capital T, capital T. And here you will get small t, small t. These are the two gametes, right? Here capital T, capital T. Then in the F1 generation, what is the possible or probable crosses here or the offsprings you can get here? Here, this capital T and small t both will join together and that will give T, T. That is the tall plant. Right, here dominant allele T is present. And here, you can see the capital T and small t both will join and that also will give capital T, small t. That also, phenotypically, that also will be a tall plant. Next, here in this condition, capital T and small t. These two will join together and it will give capital T, small t. Again, that is, this is also which one? Tall plant. Here, this capital T, the small t, both will join together and that also will give tall plant. So in F1 generation, you will get all tall plants. And in F2, if you are allowing them for selfing, see, here what Mendel did, he allowed that for selfing, right? So from the tall plant, but they were heterozygous. Both alleles were different. One capital T and one small t will come, right? The gametes, you will get one gamete will be with the recessive one gamete with the dominant one. And here, the another one also will give the same type of gametes, capital T and small t. So while forming the generation, F2 generation, you will get different combination, capital T and capital T, both will join together and you will get homozygous dominant. Here capital T and small t will join together and here you will get heterozygous dominant. Both alleles are different. And here small t and small t. So you will get homozygous recessive condition here. And here capital T and small t. So that is homozygous dominant condition. So phenotypic ratio, 3 tall plants and 1 dwarf. Right. So phenotypic ratio is 3:1 in this Punnett square. And here the genotypic ratio, genotypically when you are checking all these things, here you will get 1 tall homozygous. Genotypical ratio you are checking means 1 homozygous tall. 1 homozygous tall, then 2 heterozygous tall. These two heterozygous tall, right? Both alleles are different. Then 1 homozygous dwarf. These both alleles are similar. So homozygous condition. These both alleles are different. So heterozygous condition. Again, these two alleles are similar, but it is the recessive form. So homozygous recessive. So that's how the Punnett square, which I have explained now, that shows the parental tall character, male parent character, and the dwarf, female parent character, and the gametes are produced. And in F1, all tall heterozygous tall progenies. Then when it is moving to the F2, phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. Okay. So next is the concept of test cross. Okay, so the previous experiment and the explanation, from that you know how the generations will be, right? So it's clear that though the genotypic ratios, those can be calculated by using this probability mathematical equations and all. So by simply looking at the dominant trait, it's not possible to know that what is the genotypic condition. See, if you are getting a tall plant, you cannot say that whether it is homozygous tall or heterozygous tall character condition. The genotypic condition you cannot guess. Morphologically, it will be like a tall plant, but you can't say that whether it is both the alleles which are representing that character, both the alleles are in homozygous condition or whether it is in a heterozygous condition, right? So for example, if you're taking tall plant from F1 or F2, here the tall capital T or the capital T small t composition, both is possible, like both conditions are possible, right? So we cannot predict that. So to determine the genotype of a tall plant at F2, in F2 you want to determine that what is the genotypic composition of a tall plant or a particular character. So what Mendel did to prove that, to get the genotypic condition of the tall plant, so to determine the genotype of a tall plant at F2, Mendel crossed that tall plant from the F2 with a dwarf plant. Crossing of this F2 plant with the recessive parent. Okay, so that is called as test cross. F2 plant crossing with the recessive parent. That is called as test cross. Okay, so in a typical test cross, an organism which is showing dominant phenotype, which is having dominant phenotype, like it may be in homozygous condition or it may be in heterozygous condition, that phenotype is crossed with the recessive parent instead of self pollination. So usually in F2 what we'll do, we'll give it for selfing. Afterwards there is no cross pollination, right? So instead of selfing, there we will do cross pollination. So the progenies of that cross, that we can easily analyze and we can predict the genotype. We can predict the genotype of that test organism. Okay. See here, if you want to determine the genotype of a violet flowered pea plant, violet flowered pea plant, we want to know that. See, this is the violet flowered pea plant. Okay, so we want to know the genotype of a violet flowered pea plant, which is the test organism. This is the test organism. We are checking the genotype of this. Then that should be crossed with the recessive parent. So that should be crossed with the what? Recessive parent. Which is the recessive parent? White flowered pea plant. Okay. So how we will do? So here, homozygous recessive plant, that is crossed with the recessive parent, white flowered pea plant. right? So we want to know that this is a test organism, violet colored pea plant. So violet color, that may be due to two dominant alleles, or it may be due to one dominant and one recessive allele. So how we will come to know which one is correct? So if the condition is like, see, both are dominant, violet color, and we are crossing with the white colored pea plant, recessive, right? This will give gametes in this form, right? Both will be dominant alleles. And here also you will get recessive alleles. So how you will cross? Here you can write here the condition. The first one we are doing now. See, with the homozygous dominant condition we are checking. Here the recessive one we will write. Recessive condition. Here the dominant. In this condition, dominant, that is also homozygous. Okay, so in the first combination, you will get capital W and small w. In the second one also capital W, small w. Here capital W, small w. Here capital W, small w. Here all flowers are violet. All violet, right? Because violet dominant gene is present here. All flowers are showing violet. In the same way here, we are going for this heterozygous condition. One is capital W, another one is small w. And here the recessive, same recessive parent we are crossing, small w, right? So capital and small, in this combination also you will get capital and small, and here small and small, this small and this small w, here also the same. So half of the flowers are violet, but here all the flowers are violet. Here half of the flowers are white. Okay, so you can make out how you can interpret that unknown flower. In this case, the unknown flower is homozygous dominant. Homozygous dominant. If all flowers are violet means homozygous dominant, right? If it is in heterozygous condition, you will get half of the flower violet and half of the flower white. So we can make out whether it is in a homozygous condition or heterozygous condition. Okay. Then next is Mendel's law of inheritance. Actually, after all these experiments, based on the observations on the monohybrid crows, Mendel proposed two general rules. Two general rules. Which are they? The rules are called as laws of inheritance, or the principle or the laws of inheritance. These rules are called as laws of inheritance. There are two laws. First law, or law of dominance. Mendel's law. In that first law is called as law of dominance. First law of dominance is the first law, and second law is law of segregation. First law of dominance and second one is law of segregation. Okay. So Mendel conducted several experiments there, right? So among all these experiments with different characters, he tried with 7 characters. So in each case, he found that all characters, Mendel's experiment, he repeated with 7 traits. The same experiment he repeated with the 7 traits for confirmation. So in each one, he saw that all characters are controlled by discrete units, which are called as factors. These are factors. All the characters are controlled by factors. And these factors occur in pairs, right? All the characters are controlled by factors. And these factors are occurring in pairs. Pairs in the sense either it may be capital T, small t, or capital T, right? Or small t. See, this all these are representing a feature or a character or a trait which is called plant height. Plant height is a trait. So while we are considering about this plant height as a trait, we can guess that there will be different forms of the allele. But whatever may be, that occurs in pair. So in a dissimilar pair of factors, here you can see only one is able to express, right? Here these are the different way of expression of a character or a trait. So from this you can make out. So the third point is in a dissimilar pair of factors, one member of the pair dominates. That is called as dominant. Another one is called recessive. Okay. So the law of dominance that try to explain the expression of only one of the parental character in a monohybrid cross. In which one? In the F1. In F1, only one parental character will get expressed. And in F2, you can make out that their expression of both the parental characters you can see. And according to the law of dominance, first we need to tell that all characters are controlled by discrete unit and these units are called as factors. And these factors, then we need to tell that these factors are occurring in pair. And third point is the pair, if it is in a that is a dissimilar pair. Similar pair means there will be only one condition, right? Both factors, the factor which are occurring in pair, both the in pairs, both are having similar condition means that character without any doubt the same thing will get expressed. If there is a dissimilarity in that pair, the factor, one of the factor or one member of that pair that dominates and the other one is getting unexpressed, that is called as recessive, and the expressed one is called as dominant one. So in a monohybrid cross, the law of dominance is used to explain the monohybrid cross. So in that, in the F1 generation, only one character, one of the parental character is getting expressed, and in F2, both the characters, both the parental characters are appearing. Okay, and that explains in which ratio it is getting expressed in F2. In F2, phenotypic ratio, phenotypic ratio of the character, parental character expression, that is 3:1, and the genotypic ratio, that is 1:2:1. Okay, that's what we have learned. The next law is law of segregation. So according to law of segregation, here law of segregation means when these factors are getting segregated, or these factors, the pair that they are called as alleles, they do not show any blending. Okay, so this law is based on the fact that two factors of a character which is present in an individual, they will not get mixed up or there is no blending, and both characters, both the traits are recovered as such in the F2 generation. How that characters are there. Like that, it is getting expressed in the F2 generation. Okay. But this, both the characters will not get expressed in the F1 generation, but in F2 generation, you can see that. See, during gamete formation, that parents contain two alleles during the gamete formation. So the factors or the alleles of a pair that will segregate from each other. Okay. So while segregating, what will happen? Only the gamete, one gamete will receive only one of the two factors from pair of allele, one allele that will receive. So during gamete formation or spore formation, factors of a pair that is getting separated or segregated from each other. So one gamete will carry only one factor of a character. One gamete will get only one of the two factors. So among these two factors, one gamete will get only one factor. Okay. So that ensures, that makes sure that the purity of gametes. So of course, homozygous plants that produces all gametes, and all these gametes will be in similar condition. Okay. But when it is coming to the heterozygous condition, see, from a tall plant, this is in a homozygous condition. You will get similar type of gametes only. Capital T and capital T. Both the gametes are similar, right? But when you're going to an heterozygous parent, here you will get one capital T gamete and one small t gamete. But still it is making sure that both are, see, this is the dominant allele and this is the recessive allele. Okay. So that shows the purity of that gamete formation. Okay. So each having one factor with equal proportion. So this law, it is applicable, universally applicable law. So the heterozygous parent will produce two kinds of gametes and each are having one allele, and that also in equal proportion. Clear? So that is the two laws, like law of dominance, which is the first law, and law of segregation, that is the second law. Okay. Next, see, wherever you are going, there, in the case of science, there will be some exceptional cases, we said. Okay. So exceptions for this Mendelian principle, law of dominance and law of segregation, for that also some exceptional cases are there. So that's what we are learning as incomplete dominance. We said one character will get completely expressed, right? So here, this incomplete dominance, these are all the cases of exception to Mendelian principle. Okay. So first exception we are learning about incomplete dominance. So let's see, after learning all these Mendelian principles and all, some cases scientists observed that F1 phenotype, that is showing an intermediate character between the dominant and the recessive phenotype. Okay. So uh, scientists were studying about the Mendel's law and they were concluding, they were in a conclusion that in the F1 generation, one of the parental character is getting expressed, right? But there also they got some exceptional cases that was showing in between character, in between dominant and recessive phenotypic character. That means here in the F1 generation, it was not resembling either the male parent or the female parent, but the phenotypic expression of the F1 generation, that was in between these two. Okay, in between the male parent and female parent. So for example, here we can say incomplete dominance. You can see example in snapdragon plant, this plant. In that, Here, this is true breeding, red flower. True breeding, red flower. Both the alleles are similar, capital R, capital R. That is crossed with true breeding, white flower. This is the recessive character, smaller, smaller. So the red, the dominant character, and the recessive character, white, true breeding, white plant. They crossed these two, cross pollination they did, and the gametes formed from the true breeding, or in the homozygous condition they produced capital R as a gamete, and the homozygous recessive character small r they produced. And in F1 generation, according to Mendel's law, the F1 generation which is produced, that should be similar to one plant or the dominant plant, right? The dominant condition should get expressed in the F1 plant. But here in this case, the F1, see, according to Mendel's law, everything should be red. In all flowers should be in red color. The dominant character is getting expressed means all flowers should be in red color, right? But here all flowers were pink. All flowers in F1 offspring condition will be capital R and smaller. That is correct. But the flower color, the phenotypic expression, that was pink. So according to Mendel's law, which color we will expect? Red color, right? Here, one allele is incompletely dominant. It is dominant, but not completely. That dominancy is in such a way that is incompletely dominant over the other allele. It is dominant over the white flower, but it is not in a complete red color. It is in between. So there is an intermediate phenotype which is produced by F1 hybrid with respect to the parent, red parent and white parent, right? So if the F1 is selfing, then the F1, they allowed for selfing, right? Their selfing happened. That means capital R, smaller, capital R, smaller. While doing that, in the F2 generation, all, here you can see one capital R smaller, here capital R smaller, capital R smaller. Here what both same, small capital R, then here capital R smaller, here capital R smaller, and here smaller, smaller. That means one homozygous red dominant one, homozygous dominant red color they got, and two pink flowers, like heterozygous dominant pink. Last one homozygous recessive. Okay, so here in the F2 generation there are three types of red. Three types. One is capital R, capital R. Next, pink color, capital R, small r. And last, white color flower, smaller, smaller. But this is in 1:2:1. According to Mendel's law, the phenotypic ratio of F2 is 3:1. 3 red and 1 white color should come. 3:1 it should be. Right? But here it is showing 1:2:1. So that's how the incomplete dominance. So in the ratio 1:2:1, this is Mendel's law. Mendel's F2, right? So in the heterozygous condition, capital R and small r, right? In this condition, that phenotypic effect on the allele, that is more pronounced than the other one, and there is a mixing is occurring. Here, a mixing is occurring between the red and white, and as a result of that mixing or blending of character, their pink flowers are developed. The flowers are developed in a pink feature. Okay, so here genotypic ratio is 1:2:1 and the phenotypic ratio is 1:2:1. So in incomplete dominance, you will get 1:2:1. Both the phenotypic and genotypic ratio, both are similar. Okay. So about the dominance concept that we said, like all genes are having some information. That's how that character or feature or a particular trait is getting expressed. That gives the expression. So in a diploid organism, two copies will be there for each gene. So that pair or that copy, that is called as allele, and this allele, we cannot say that always these alleles will be in an identical condition. If it is homozygous, it will be identical. But heterozygous means, as we said, it will show difference, that alleles. So one of them may be different due to some changes. So the information that particular allele is having, that also may change. Okay, so if a gene is having some information to produce a particular enzyme which is expressing that character, that in the two allelic form, the normal allele will produce normal enzyme which is needed for the activities, for the expression or the transformation of that substrate. Okay, so there is a normal allele which is producing a normal enzyme, but the other one, due to some condition, that might have changed. So there is a modified allele which is responsible for the production of the uh, normal enzyme. Sometimes it may be producing the normal enzyme. Here in the case of that incomplete dominance, we said like it is producing pink color, right? Instead of red, it is giving pink flower. So why? Both the alleles may be similar, but due to some condition, that second allele, one is producing the normal enzyme, but the other one which is producing the normal enzyme, that may not be efficient. Or sometimes the other allele, the second allele, which may not be functional, it may not be able to produce a functional enzyme. And sometimes there won't be any enzyme at all. So these are different conditions, which is a reason for incomplete dominance. Okay, there are several reasons. Sometimes that part may not be functional. Sometimes it may be functional, but not that with that much efficiency. Okay, sometimes that part or that allele may be non-functional. So these are some reasons which we can say for the incomplete dominance. Okay, then ABO blood group in human being. This is the codominance. This is showing the codominance. See, in human being, we can say that codominance, the F1 generation that resembles both parents, F1 generation. So here the best example we can explain for codominance, that is the blood group. Different types of red blood cells, which is determining the ABO blood group in human beings. Okay, so this ABO blood group, that is controlled by which gene? I gene. Gene I, that is controlling the ABO blood group. So the plasma membrane of the RBC cells, that is having sugar polymers, which is protruding, which is showing or protruding from the surface. In the plasma membrane, RBC plasma membrane is having some kind of protuberance which is from the surface, and that they are the sugar polymers. Okay, so the genes, the kind of sugar which is controlled by the gene. This gene I, that is having, I gene is having 3 alleles. Okay, 3 alleles. Which are the 3 alleles? These 3 alleles are IA, IB, and recessive I, a small I. So the IA, the allele IA and IB, these both are slightly different from each other because of the sugar. But the small I, IA and IB, these are slightly different sugar forms. These are slightly different form of the sugar, right? The polymers that protrude from the surface, sugar polymers, that is different in IA and IB. It is showing slight difference. That's why we are calling IA, and another type is IB. But the small I, that is no having sugar polymer which is protruding from the surface. Okay. So here in human, each and every person, they'll be having any of the 3 I gene alleles. We said IA, IB, and small I. So all human beings, they'll be having any of this gene. Okay. Any of the 3 genes will be there. And this IA, that is dominant over small I. Small I is the recessive form. And IB, that is also dominant over small I. Okay. We can say that IA and I, that will give, if both are coming together, that will give only IA expression. And when IB and small I both are coming, that also give IB expression. Like IA and small I, this will give IA expression only. At the same time, IB and small I both are coming means that will give IB expression. Okay. But when IA and IB both are present together means both will get expressed, or both are giving their own expression. That is because of codominance. So in human blood group, codominance, which one you will say as an example? AB, IA and IB both are coming together means both expression, both will get expressed. Both will give the expressions. So red blood cells that have both A and B type of sugar. This condition coming means that RBC will be having A type and B type sugar. Okay. So there are 3 different alleles and there are 6 different combinations of these 3 alleles which are possible. Okay. So total 6 different genotypes of human ABO blood types are there. 6 types, different genotypes of human blood groups are, or ABO blood types are present. Okay. For codominance, we will say both A and B, both are present. So these are the blood groups in human population. You can see here, from parent 1, it is getting IA allele, and from parent B also IA allele. Then in the offspring, both IA and IA will get expressed. This is homozygous condition. So that offspring will be having A group, blood group will be A, right? And next, IA and IB. From one parent it is getting IA, and from another parent it is getting IB. Then both will get expressed. This is the codominance, example for codominance. So they will be having AB group. Okay. Next, from the parent 1, it is getting IA, and from parent B, it is getting the recessive allele. Then IA will dominate over the recessive allele. That time also, offspring will get A group. In the same way, IB from first parent and from second parent, IA is coming. Here also, both will get expressed, and that offspring will get AB group. Here, IB and IB, from both the parents, IB are coming. Then offspring will be having homozygous dominant condition of B. that means B group. And when IB from one parent and recessive allele I from another parent, that time IB will dominate over the recessive allele and that parent, offspring will get B group. Then in both, from both the parents, they are receiving recessive allele, means there won't be the presence of any dominant allele. Both recessive allele, it will receive from both the parents and that forms the O group. Okay, so here codominance AB group, that is the example for AB blood group. That is the example for codominance. Okay, and next comes the concept of multiple alleles. Multiple alleles. So that also we can explain with the help of what? Which one? We can explain with the help of human blood groups. Okay, here the same thing we have explained. So from one parent IA, another parent also IA. Both the alleles are coming and that forms the A blood group. Next IA and IB, that forms IA IB condition in the genotype, that gives AB blood group. Then IA and small I, that gives dominant IA condition, that gives A blood group. Then here IA and IB genetic condition is coming, which again it is giving AB blood group. Then both dominant condition IB from both parents, that gives B blood group. One dominant allele IB and another one recessive small I, that is also giving B blood group, and both in recessive condition means that is giving O blood group. So here different types of RBCs that determining the ABO blood grouping in human being, that is controlled by one gene, I gene. So the plasma membrane of this red blood cell, that is having sugar polymers, which is polymers, which is protruding from the surface, and that which type of sugar is present, that is controlled by that gene. So the gene I, which is having three alleles IA, IB and small I. Okay, so all the individuals which are having 2n condition, they will receive only two alleles. So the multiple alleles can be detected only in a population. In a population, we can detect the multiple alleles because there are three different alleles. Therefore, six different types are possible here. Right? Here you can see six different types. So for this character, many phenotypes are possible. Here you can see many phenotypes. Genotypically, we can write this. So phenotypically, these many possibilities are there. So we have to check with this multiple allelic conditions, right? So different types of allele, which are the three different types of alleles? IA, IB and small I. These are different alleles which is produced by the same gene. Which gene? I gene. Different forms of that allele. So this is a concept of multiple allele. Then another example you can see in multiple allelism, that is a starch synthesis in pea seed. Actually, this is starch synthesis is controlled by one gene in pea plants. So that is having two alleles, B, capital B and small b. So starch, how it is getting synthesized? When this B gene, it is coming in homozygous condition, starch synthesis is occurring. So large, that gives large starch grains. Okay. So when it is coming in homozygous recessive condition, that gives lesser efficiency in starch synthesis. So small starch grains will come. Okay. So after maturation of the seeds, when they are in homozygous condition, they are round. These will give round condition. But when they are in homozygous recessive condition, that will give wrinkled seeds. Okay. When heterozygotes are getting produced, so that time this capital B, that is the dominant allele. But the starch grains which are produced when it is coming in intermediate condition or heterozygous condition, here dominant allele is there, but along with that recessive allele is also present. So that time intermediate size you can see in the seeds. Intermediate size of seeds you can see. Okay. So here you can see incomplete dominance. The dominance is not complete. Okay. Then how two genes are getting inherited? See, then now monohybrid single character, whichever Mendel is followed, that's over. Next is with the cross with two genes with two different characters he concentrated. And here you can see the pea plant parental generation. He considered round and yellow color, round seed with yellow color. And here next opposite parent, wrinkled seed and green color. Two characters he considered. Okay, this round, that is a dominant character, and for that the opposite or the recessive character is wrinkled. And here yellow is the dominant character, and for that recessive character is green. Okay, so while mentioning round and yellow, when we are mentioning they're wrinkled and green, you should mention with the same small letters only. Okay, so how the gametes will be round and yellow, round and yellow. This is one gamete, and from here wrinkled and green. This is another gamete. So in F1, how you will get round, this is how in F1 it will be. For round, these are the two alleles, and these are the two different alleles of the same gene. So R, capital R, small r, capital Y, small y. But here both the dominant alleles are present. So the phenotype, how the expression will be round and yellow. Clear? So Mendel worked with the cross pea plant that is different in two characters. Round color, that is dominant over green color, and round shape, yellow color, which is dominant over green, and round shape, which is dominant over wrinkle. The same thing. Then here you have seen the F1 generation. When it is moving to the F2 generation, you will get different types of gametes, right? From the female, you will get these many gametes. Capital R, capital Y, small r, capital Y, capital R, capital R, small y. Here also the same thing will get repeated. So by using the Punnett square, if you are making, you will get a phenotypic ratio, not genotypic, phenotypic. Genotypic condition you can see in this columns, right? So these two are joining, two capital R, two capital Y, right? How it will be? This will be round and yellow, right? So all these, when we are checking with the phenotypic character, you will come to know that the phenotypic ratio is in such a way that 9 is to 3 is to 3 is to 1. That means round and yellow 9, then round green 3, then wrinkled yellow 3, wrinkled green 1. So like that, you will get different combinations when you're matching all these things and writing. The genotypic condition you can write in the column, and phenotypic condition you can see here, right? When you're writing each one. Okay. So this is the inheritance of two genes. So when we are making a cross between pure round yellow seed pea plant with wrinkled green seeded pea plant, yellow color which is dominant over the green color and round seed shape is over the wrinkled seed shape. Okay, so here RRYY, that you can see only one, that is the pure round and pure yellow, right? Next, if it is in RRYY condition, this is pure round and hybrid yellow color, one capital Y and one small y, both are there, no? So the pure red and hybrid yellow, like that you can write about each character. When it is coming RRYY, these all are recessive genes, means wrinkled green. See this one, this condition, wrinkled green. This is hybrid red plus which one? Green seed, right? Small y represents the green color. Okay, so this is the result of a dihybrid cross when two parents with different two pairs of contrasting trait. So here we are taking seed color and seed shape as the contrasting traits. Okay, so that's all about this class, about the inheritance, way of inheritance we have learned today. So just practice with different traits, contrasting traits, the monohybrid and dihybrid cross. You can, monohybrid is very easy, but dihybrid with two contrasting features, you have to write this and practice. So it will be easy for you to solve the problems. Okay, thank you.